Hi everyone, it's 4 o'clock here in the UK, so we'll get going on the next SimSip scientific webinar. Thanks for joining. If you could remain muted throughout the course of the presentation, and we'll have questions at the end, but please also feel free to write them as you're going along in the chat box. And just a note that we're recording the presentation today, and it should be available on YouTube in a couple of days or so. So yeah, our talk today focuses on biologics, and it's Dr. Felix Steder. He's a associate principal scientist at Certara Predictive Technologies, so he's within the SimSip group. He did work with us prior to doing his PhD, which he did at the University of Basel in medical biological research, and he has been heavily involved in development of biologics simulator as well as involvement in populations. So you might have seen publications of his in that field as well. So today's paper, and the reason we asked Felix to present this, it was a recent publication. It shows, or at least showcases, some of the collaborative work that can be done within the focus groups, in this case the biologics focus group, and it's specifically focused on looking at modeling in terms of case studies across translation from monkeys to humans, and yeah, looking at gaps in the field. So we appreciate you putting together this slide deck, obviously to present the paper, looking at the different case studies and the learnings that you drew from that. So otherwise, I'll pass over to you, Felix, and thanks again.
Thank you very much for this kind introduction, and welcome you all to the scientific webinar, where indeed we will talk a bit about modeling of biologic case studies, but we will also look in if we need a more mechanistic clones modeling for therapeutic proteins. And I will explain the sentence a bit during the presentation, especially towards the end, to what we mean, which mechanisms we should focus on and which mechanisms we should have a look at. As Ollie said, this was the work of a cross-industry collaboration. So 4 years ago at the SimSip Consortium, we founded the Biologic Focus Group. And the aim was indeed to bring different industry people together with case studies of therapeutic protein simulation to get a better understanding of about model verification, as well as the need of a standardized workflow and the need for standardized parameters that are required. We had some loss to follow-ups in this group due to various different reasons. So the people that I've listed here are the co-authors of the paper that have finally shared their case study examples. You can also see that the entire biologic group of SimSip has been involved in this work in terms of providing guidance, supervisions, and also an own case example that we have used. So I'd like to thank all of the people for their hard work here. Okay, let's get started. Because I said we are talking today about biologic case examples, but what do we actually mean by biologic drugs? And the pie chart here on the left-hand side shows the summary of approved biologic drugs at the FDA in the time frame of 2020 to 2024. What we see there is typically an approval of 10 to 20 biologic drugs per year. And what you can see from this pie chart is that the majority are monoclonal antibodies of almost 60%. If you go further in the pie with the orange and the gray part, we can see here antibody drug conjugates. ADC in short, and bispecific antibodies. So antibody-related products are more than 2/3 of the entire approved drugs. So they play a dominant role here. And if I ask someone what is a biologic drug, it's probably monoclonal antibodies is the first that comes into mind. But it is really important to realize that biologic drugs are more diverse. We also have peptides, proteins, enzymes, hormones, and we also have oligonucleotides, antisense oligos, as well as siRNAs. And the important point is that all of those moieties can be simulated by a PBPK model. So what can we use modeling and simulation for when we think about biologics? One of the first things could be the identification of trackable targets. What do we mean by that? If you have a drug candidate, a biologic drug candidate can be a monoclonal antibody, but can be any other protein or an oligonucleotide, you have a certain target. What we can do by using the characteristics of this drug is to ensure very early in drug development that under physiological conditions, sufficient drug reaches the target, you get sufficient target engagement, and therefore a likelihood of having an effective drug. What we can also do is to look at the link between molecular characteristics and the pharmacokinetics. That means what if questions. What if we change binding affinity to the target? What if we change other characteristics of the molecule? Because we can still predict under physiological condition what happens at the target site. How does the target engagement changes? How much of our drug actually reaches the target? We can predict concentration at the target site in general, but we can also look at off-target effects. And this is also very interesting thinking, because if we start injecting any of the drug subcutaneously, which is one of the preferred routes, and we start to see skin lesions, then it could be that target, for instance, is also expressed at the skin site. And we have to consider the target engagement at skin, although the desired actor is maybe in a tumor to fight against cancer. And knowing the level of the target and knowing the location of the target and using a PBPK model helps us to understand where target binding happens, where target engagement happens, and how it supports the explanation of those off-target effects. We can also look at the bioavailability after subcutaneous administration and predict this a priori. We can look at appropriate doses in so-called special populations. That means populations with limited clinical data. And we can go to the bread and butter of SimSip, and we can look at the prediction of drug-drug interactions. Those can be cytokine mediated, but those can also be the interaction between a released payload of an antibody drug conjugate and any co-medication that is given to the patient. What is very crucial is to understand the physiological processes before we can develop a mechanistic model. So what I've put in here is the physiology, the physiological processes that happens for antibodies. As you have seen, they are the majority of the biologic drugs that are approved and that are currently worked on. But these processes are not just for antibodies. So all proteins and partially also oligonucleotides undergo those processes. So what we have after drug injection is that we have high concentration of the drug in the blood, in the systemic circulation. There are two ways for proteins and oligonucleotides to distribute from the vascular space, from the blood vessels, into the interstitial space. One is paracellular. So what we have between what we have is that endothelial cells surround the blood vessels. And there are fluid-filled pores through these endothelial cells that connect the vascular space and the interstitial space. And we can have paracellular distribution of those larger molecules between the vascular and the interstitial space. The other distribution pathway that we have is shown here on this slide. We can have pinocytosis uptake into the cells. Pinocytosis is unspecific in a way that the cell takes up surrounding fluid and all molecules that are included in the fluid distribute into the cell through pinocytosis. So there could be endogenous molecules such as albumin or IgG. There can be exogenous administered molecules such as antibodies, proteins, or oligonucleotides. After pinocytosis, the drug is in the early endosome. We start to see acidification from neutral pH down to a pH of 6. And there's a receptor in those endosomes, FcRn. And all proteins with an Fc domain, or albumin and albumin fusion proteins, they bind to this receptor under acidic conditions. And the complex is then recycled back to the blood. Or there can also be transcytosis into the interstitial space, although that seems to be less likely compared to the recycling back to the systemic circulation. And this recycling pathway leads to the very long half-life that we see for IgG and antibodies. All molecules that are unbound, and that are also proteins, peptides, oligonucleotides, they can be degraded. For oligos, there is another pathway that is not listed in here. They can escape the endosome and be released into the cytosol and then have their action in here. So you can see that those processes are known, and we can include those into the model. So here I show the model that has been used. On the left-hand side, we see in all of the cases that are presented today, a full PPK model has been used. And this is really independent of the molecule type you look at, because we always have a lung, liver, or kidney independent if it's a small molecule, a protein, or an oligonucleotide. What we include for larger molecules is the lymphatic system, and this is shown here as the orange dashed lines. These are the lymphatic flows from the different compartments that are collated into a central lymph node compartment and then brought to the venous bloodstream. On the right-hand side, you can see the generic drawing of a compartment. Because of the size and the polarity of the molecules we are stimulating, we require a permeability-limited structure. So each of the compartments is separated into the vascular space, representing the blood vessels, the endothelial cell layer that surrounds the blood vessels, the interstitial space, and the intracellular space. And you can see some of the processes that I've talked about in the previous slide that are included in here in a more technical drawing. So we see two-pore distribution, that is the paracellular pathway between the vascular and the interstitial space. We see endocytosis and recycling, and we see that FcRn binding can occur in those endothelial cells. Target binding can also be considered as for membrane-bound targets and soluble targets, but plays less of a role for this presentation, as you will see later. What are inputs that we require? One of the parts are the physiological data that are already included within the SimSip simulator. So those include body height and weight, tissue volumes, and blood flows that we need also for small molecule drugs. But for biologic drugs, we require more data, because I already said that we have to include the lymphatic flow. So we have to include the lymphatic system lymph flow as well as the central lymph node compartment volume. For monoclonal antibodies, we also require IgG concentration, the endogenous IgG concentration, and the abundance of the receptor in the endosomes, the FcRn abundance, and of course target abundance if target binding should be stimulated. Physiological data can come from animals and can come from humans. In terms of drug data, it is simpler for biologic drugs if you're used to small molecule drugs, because what we require in all cases is hydrodynamic radius. Hydrodynamic radius of the molecule is used to calculate the two-pore distribution, the paracellular distribution pathway. For all other parameters, it depends a bit at the molecule type we're looking at. So if it's a protein, peptide, or oligonucleotide, the fraction unbounded plasma becomes important. For monoclonal antibodies, they don't bind to albumin, so their fraction unbound would be always 1. But monoclonal antibodies require binding affinity to the FcRn receptor. What is also important is to have an idea about Clowen's pathways and, of course, binding affinity to the target. So what's the workflow that we think of in developing a biologic PPK model? Ideally, we start from in vitro data to inform the model to build up a true bottom-up model. Then we can develop and verify models in preclinical species. So that can be rodent, that can be the monkey. We can also go from one species to another. But the final aim is then to extrapolate to humans from either of the species, or from rodents or from the monkey. And the beauty of PBPK modeling is that we are not finished by this at this part, because it takes some time and work to build those PBPK models. But once they are fully verified, they can be used for different application scenarios. So we can not only do the first in human dose prediction, but we can also look at DDI potential. We can also look at different patient populations. We can, for instance, also look at the drug exposure in children. This was a general introduction a bit into the PBPK models that we're using and what can be done with them. So now over to the objectives of the current study. As I said before, we formed a cross-industry collaboration, and the aim was really to collect cases for different biologic drugs. The aim was, or we had three aims. The first aim was we wanted to evaluate a standardized workflow. We wanted to get an idea about standardized model parameters, and we wanted to gain more confidence into the model in terms of verification when we look at the observed risk prediction for different biologic drugs. The case study could have been any therapeutic protein, independent of size, route of administration, or elimination route. We restricted it to therapeutic proteins at that time because we did not have the PBPK model for oligonucleotides. That was included 2 years ago. So that could be a potential approach for another biologic working group. We required to have clinical data in humans. That was an absolute requirement. And additionally, we also asked for preclinical data in the monkey. And at least one dose had to be high enough to saturate the target. So in the end of the day, we had 7 different case studies. So you can see that 5 of those case studies were monoclonal antibodies, 1 was an antibody drug conjugate, and 1 was a bispecific T-cell engager. So the distribution is pretty much what we have seen from the approved drugs from the pie charts. The majority are monoclonal antibodies or antibody-related products, and only a minority is a different one, is a different biologic class. The route of administration was mostly IV, apart from the bispecific T-cell engager that was administered subcutaneously. We always had data in humans because that was a requirement, as I said. And in half of the studies, we had also data available in the monkey. Very importantly is one case study is not necessarily just one drug at one single dose. So one case study could have different doses of the same molecule, but it could also be different derivations of the molecule. So in terms of the bispecific T-cell engager, there was a couple of different derivations of the molecule that were all included in a case study. So in the later figures, you might see different circles in there. This was the workflow approach that we agreed on in this collaboration. So we started with in vitro measurements and the binding affinity to the Fc receptor, because 6 out of 7 case studies we had antibody and antibody-related products. We then developed a PBPK model in the monkey for those studies who have monkey data, especially looking at the species-related differences between the human and monkey receptor. And then we built the model in humans for all of those. And here we looked at different parameters, amongst other, the additional plasma clones. So what are the input parameters for the different drugs? In all cases, we used molecular weight, and molecular weight is used to predict the hydrodynamic radius, which I said is important to estimate the 2-pore distribution through the paracellular pathway. And this is relevant for all biologic drugs. The binding affinity to FcRn was important for monoclonal antibodies and ADCs, but is not required for the bispecific T-cell engager. Bispecific T-cell engager was small enough to be renally filtered, so we included renal filtration clearance. The additional systemic clearance was required for all drugs. This is very important. And deconjugation is only relevant for the antibody drug conjugate. If a parameter is not listed in here, for instance, the endocytosis uptake rate, then it was unchanged compared with the SimSip default. So our first aim was simulation of antibodies in the monkey, but as you have seen, we only had data for 3 antibodies available from our case studies. So we went into the literature to find 14 additional studies for humanized IgG-like antibodies in cynomolgus monkeys that were administered IV and were said to have no TMDD or high enough doses to saturate the target. And this was done to prove the simulations in the monkey. What we tested in a monkey was whether we can use minimal input parameters. So they are molecular weight, binding affinity to FcRn, and additional systemic clearance of IgG. Very importantly is that the binding affinity of humanized IgGs is different to the human FcRn receptor and to monkey FcRn. So in an ideal case, we said in the workflow that we start with in vitro measurements to this receptor. But in all case studies, the measurements was only done to human FcRn. So one of the questions was, do we need the measurements to monkey FcRn as well in order to build a reliable preclinical model? What we did in the first instance was we took a measured relationship for IgG that was published 10 years ago by Abdish et al. What they did in this study was they used IgG from different species and they used FcRn from different species and they measured the binding affinity. So for humanized IgG binding to monkey FcRn, we see a KD value of 332, and humanized IgG to human FcRn has a KD value of 737. So there's a factor of 0.45 between them. So we had the measurements to human FcRn, and we applied this factor of 0.45 to all of the case studies, and evaluated whether measurements to human FcRn is sufficient if we're multiplying or we're using this factor that has been published. This is the result of the plasma concentration that was predicted in the monkey of the different literature examples. In all cases, monkey was non-cross reactive or the dose was high enough to saturate the target. So we dose normalized to 1 mg per kg in order to make the data comparable. You can see here the black line is the SimSip prediction. It's only a single population representative for the 4 kg monkey that was simulated. And the red dots show the observed data. So you can see we have a pretty good fit of the data. Half-life was around 12 days, which is supported by literature data. What we then did is to look at the Cmax and the AUC, and this was the prediction error between the predicted and observed values. We asked the Biologic Focus Working Group for their drugs to provide us with the prediction error for Cmax and AUC in all cases, because what we wanted to have is not like focus on an individual drug with all the individual data, but rather have a bird's view and get a more general understanding of this. So the biologic cases from the 3 different case studies are shown here as the red crosses, and the literature example are here in black circles. So what we can see here in general is a good prediction of the Cmax to the left-hand side. We see one of the drugs is outside of the 50% interval that we decided on at the beginning of the group. You could argue that there might be a slight tendency of over-prediction of Cmax. I wanted to say here that all of the case studies here have been administered IV. So after intravenous administration, One of the factors that is very important is plasma volume, and plasma volume highly depends on body weight. And we did not use the individual body weight between the different studies. We always used 4 kg in the SimSip monkey simulator. So that could be one way of refining it a bit better, but we are absolutely within the range here. And you see on the right-hand side is perfectly well predicted, and there seems to be no bias towards over or under prediction that we're seeing here. So that concludes the monkey simulations to say, yes, we can use minimal parameter input, we can only use molecular weight, and we can use measurements to human FcRn by using this factor that has been published by Abdish et al. The second part was the simulation in humans. And there we run 3 different scenarios, specifically focusing on the additional systemic clearance. Scenario 1 in green on the left-hand side was no additional systemic clearance, so the parameter was put to 0. In the middle in yellow, there is the additional systemic clearance allometrically scaled from the monkey, in case for those 3 case studies where we had monkey data. And for all cases, we fitted it to the highest available dose. That was a requirement for all the case studies. So this is looking at the Cmax ratio. So again, on the left-hand side, we have no additional systemic clearance, in the middle allometrically scaled, and on the right-hand side, the optimized additional systemic clearance. And what we can see here is that there is no differences for the 3 scenarios. And this is actually what we expect, because most of the studies have been given IV, and the additional systemic clearance will not have any impact on the Cmax ratio in here. But there is quite an impact of this additional systemic clearance term on the AUC, because for some of the case examples, we really see a misprediction when no additional systemic clearance is used. And if you do allometric scaling or the optimized one, then we, of course, we are within 50%, especially if you do the fitting of the data. For the allometrically scaled one, we unfortunately had only 3 case studies, so it is difficult to draw a conclusion, but we definitely see an improvement using this. But the best fit is obtained if we're using an optimized clearance in here very clearly. So to sum up the results of this is, as I said before, minimal data were sufficient to predict the PK of monoclonal antibodies in the monkey. The molecular weight is sufficient, and the measurement of KD to human FcRn is sufficient if they scale to the monkey. And we can use additional systemic clearance of IgG. In terms of the human simulation, we saw that additional systemic clearance is required across the board for all the investigated case studies, no matter if it was a monoclonal antibody, the ADC, or the bispecific T-cell engager. What we have seen is that ideally, the additional systemic clearance has to be fitted. We cannot leave it out. It is a required clearance term. And this is pretty much a limitation for any de novo biologic studies, because if we require fitting, we require observed data first. And this sets up quite a limitation for using PBPK models there. So this is coming back to my original sentence. We need to have a more mechanistic understanding of this therapeutic protein clearance. That is an absolute requirement. And there are there's one part that needs to be investigated, because currently most PBPK models that are published in the literature say that elimination only happens in endothelial cells. And this is not true. Elimination also happens outside of the endothelial cells. And we have used here an additional clearance term, a plasma clearance term, that is 20% of IgG to reflect any non-FcRn dependent clearance. But those pathways are, as I said, generally neglected. So there are 2 ways how to look at the additional systemic clearance. One part are antigen presenting cells. The other part are non-FcRn expressing cells. And I will go a bit more into detail into both of them. First, we're looking at antigen presenting cells. So what is the physiological pathway there? The physiological pathway is very similar to what we have seen for endothelial cells. So we have pinocytosis that takes up fluid with all molecules that is contained into it. And those cells are highly pinocytotically active. That means in an in vitro measurement using horseradish peroxidase as the pinocytosis marker, APCs had an 8-fold higher uptake rate compared with endothelial cells. We also have the endosome, we have acidification, and FcRn is contained in antigen-presenting cells such as macrophages or dendritic cells or the Kupffer cells. The complex again is recycled and the rest is degraded. So they contribute to the homeostasis of IgG. The question is, how much do they contribute? And there was a nice study published by Richter et al. About 7 years ago in mice, where they used 4 different cohorts of mice. The first cohort were wild-type mice, where FcRn was expressed in hematopoietic cells and endothelial cells. Half-life is around 7 days after IV administration. Cohort 4 was a global knockout of FcRn. Half-life decreased about 10-fold. And cohort 2 and 3 are the interesting ones, because in cohort 2, there was no FcRn in hematopoietic cells, and in cohort 3, there was no FcRn in endothelial cells. And what we can see from the plasma concentration time profile, as well as from the half-life, both hematopoietic cells as well as endothelial cells contribute to the FcRn salvage pathway. So in both cases, we see a decrease of the half-life to around 1 day, which is not as dramatic as in the global knockout, but still significantly lower than in the wild-type animals. So the hematopoietic cells contribute to the overall IgG homeostasis, which might need to be considered in the models. The other pathway are non-FcRn expressing cells, because if you think about this, the proteins or the oligonucleotides, they are in the systemic circulation after the administration. So they are highly exposed to endothelial cells, and endothelial cells take them up by pinocytosis. But what I said is that you have the paracellular distribution pathway. So you have the distribution from the blood space into the interstitial space through those fluid-filled pores. So the interstitial space has drug concentration. And you have the intracellular space that is represented by different cell types, adipocytes, cells in the heart, kidney, whichever compartment we look at. All of those cells have the ability to do pinocytosis. That means we also see pinocytosis of those other cell types taking up the fluid of the interstitial space. And if there is protein or oligonucleotide included in there, they will be taken up into the intracellular space in here. But this is something that we currently don't consider. Of course, the concentration in the interstitial space will be lower compared with blood, so those cells are not as highly exposed to the drug as endothelial cells. So we don't expect the same uptake rates, but it might be sufficient to explain some of the catabolism that is seen. If those cells do not contain FcRn, that means also for monoclonal antibodies that they will be degraded because there is no recycling pathway. Now the concentration in the interstitial space highly depends on those pores, on those numbers of pores and the pore sizes and the structure of the blood vessels. So in some of the tissues or in the organs, such as liver, we have a very high distribution from the vascular into the interstitial space. So we will see a high concentration or higher concentration of the drug in the interstitial space of the liver compared with other organs or tissues that have tight junctions, such as muscles, for instance. Hepatocytes also contain FcRn, so there is already recycling by the body that would need to be considered as well. But other cell types are FcRn free, and therefore we would see clones there. Okay, but what are the current limitations of adding these factors? And one of the challenges that we will have is parameterization of the models. And that starts if we're looking at those antigen-presenting cells, because the question would be, how many antigen-presenting cells do we have per compartment? And this is not a static number. This is something that can change and that can be quite dynamic. And this is something that we can hardly measure. So this is one of the challenges that we will face there. And the other part is about the abundance of the FcRn, because FcRn abundance is typically measured in total tissue. So that the data that we're using is total tissue data. We don't know how much is for the endothelial cells versus any antigen-presenting cells that is in the tissue, or in other cases, such as the liver, how much is in hepatocytes versus the Kupffer cells versus the endothelial cells. So this is something that is also unknown there. And the other part is the in vitro endocytosis uptake rate. Because we don't only need the uptake rate into endothelial cells, we would also need this uptake rate into all the other cell types. Now, in vitro, as I said, it can be measured by pinocytosis marker, and this is probably quite accurate because what I said is that they take up the surrounding fluid with all molecules included in. So it doesn't really matter what the size of the molecule is there. But the question is, how well does the in vitro endocytosis uptake rate correlate to the in vivo situation under physiological conditions? What we also know is that very likely drug characteristics also play a role when we think about this pinocytosis pathway. And this can be electrostatic interactions. If you think about the cell membrane, it's negatively charged. So if you have a positively charged protein, you have a higher likelihood of this protein to getting close to the membrane, and therefore a higher likelihood of pinocytosis uptake into the cell. Now, there is currently no quantitative relationship between electrostatic interactions or charge effect and the uptake into cells. We know that very likely charge patches at the antigen binding site of antibodies play a role, but as I said, the quantitative relationship is missing. There can also be hydrophobic interactions. There can also be glycosylation that matters because we have certain receptors where the glycosyl can bind to. The ASGPR in the liver would be one example there that we also use for the oligonucleotide model. So why I'm going so into detail of this pinocytosis, and this is essentially because this is really the bottleneck. So if you think about uptake into any cell, endothelial cells, antigen-presenting cells, and other cell types that do not contain FcRn, it always depends on the uptake by pinocytosis to get into the cell and be finally degraded in the cell, because the degradation will take place in the lysosome of the cell. So the current limitation that we see here is that we would need to rely on fitting, and that could potentially lead to identifiability issues because there's just too many parameters that we need to look at. Anyway, how could an updated model structure look like? So this is a bit drawn in here, again, as a technical drawing into the current model structure, where the new parts are introduced in yellow, because we have those antigen-presenting cells in circulation, monocytes, for instance. We have those antigen-presenting cells also inside the cells. They are especially in the lymphatic system, because the task of the lymphatic system is to filter fluids efficiently from foreign substances. And this is something we have introduced into the subcutaneous absorption model and to predict bioavailability in here. And we also can include endocytosis into the intracellular space with consequent clones in there. So overall, we're getting to a more physiologically relevant compartmental structure, of course, at higher computational cost and at higher parameterization. But it would be very interesting to look into this, because if that can partly explain the additional systemic clones, then we get a better prediction, a better handle of de novo biologics, and we would no longer rely on the fitting of this clones pathway, but understand it more mechanistically. So to conclude, what we have seen in this cross-industry collaboration is that monkey simulations can be done with minimal parameters, that is molecular weight, that is binding affinity to human FcRn for antibodies, and scale it to the monkey similar to IgG and the additional systemic clones of IgG. What we have also very clearly seen across the board is that additional systemic clones is an absolute requirement. Leaving that out can lead to a high misprediction, But what we currently see is that fitting is definitely required, and that means observed data. So what we require is a more mechanistic understanding of this additional systemic clearance pathways, and that can be introduced by looking at antigen presenting cells, and by especially looking at cells that do not express FcRn and the uptake into those cells. And with that, I like to thank you for your attention and happy to take any questions.
Thank you, Felix. I appreciate you taking the time to go through that. That was really good. There have been a few questions on the chat. So if we go back a bit, it's around the additional systemic clearance parameter and where it came from. Was this calibrated to monkey data?
The additional systemic clearance in the monkey,
yeah,
that was just simply assumed to be 20% of the systemic clearance of IgG. The reason why we're using 20% goes back to an analysis of antibodies that were engineered to have higher FcRn binding. So what people thought is, if FcRn was responsible for the half-life of IgG, and we want to have molecules with a longer half-life, we just manipulate the binding affinity to FcRn, and we achieve this. And this is not true. And what you can see if you look at all of those data from preclinical species and also from the human is there is a certain threshold. You can increase the half-life by 4 to 5-fold. If you start engineering further and further and further, you won't see any differences in the half-life. And this came, the conclusion of this is there must be a clearance that is independent of FcRn. And the analysis by the PBPK model of those data led to the introduction of this additional systemic clearance at that time, and that was set to 20% to recover this 5-fold change. And this is what we have used in the monkey.
Great, thank you. And in terms of the monkey predictions, there's a question around the 0.45 factor. Was that used from human conversion, or did you have some monkey data directly from literature examples? Yeah.
No, so as I said, in all of the cases, there was only measurement to human FcRn, measurement to monkey FcRn, or also to other preclinical species was never done. So the only part was this paper by Abdish et al., where they really went across species between IgG molecules and FcRn. So we applied the factor that they've published for all of our molecules, and it appears to work. But we don't have any independent verification where in one part binding has been directly measured to monkey FcRn.
Great, thank you. There's another question around the relationship with biological phys-chem parameters. Was there a relationship to intracellular clearance?
So, okay. So by this, you mean something like those electrostatic properties, something like charge of the molecule and the effect on the intracellular clearance. As I said, the bottleneck of all these processes is pinocytosis uptake, because the catabolism of those proteins happens inside the cell. So the protein has to distribute into the cell in order to get to the proteases and be degraded there. So I am not aware that there has been any investigation between those properties of the molecule and the protease clearance, because that is quite unspecific and it has very, very high capacity. So I know it cannot be saturated under any experimental conditions that have been tested. But the bottleneck is really the uptake. You have to get into the cell. And what we know is that negatively charged proteins have a lower clearance, and the idea is that they are not getting close enough to the negatively charged membrane in order to get into the cell and be degraded. Does this answer the question?
Yeah, I think that's really clear. Thank you for that. So in terms of the internalized antibody into the cells that express FcRn, the question is, are they 100% recycled, or is there still some percentage which is like lysosome degradation?
There can be, yes. So, I mean, I said it a bit simpler because I just said every molecule that binds to FcRn can be recycled, the rest is degraded. This is under physiological conditions not extremely true. You need actually a 2-to-1 binding in order to have efficient recycling. 1-to-1 can still be degraded. And there is not 100% recycling. So if you're looking at endogenous IgG that circulates in our blood, it has a half-life of around 21 days, and then afterwards it's degraded. And it's really interesting because the cell must somehow distinguish between the IgG molecules that are aged enough to be degraded and the IgG molecules that are recycled. And as far as I know, this has not been found out what the reason for this is. But there's definitely a kind of a sorting process to sort out those aged IgG molecules. So the same happens, of course, then also to IgG-based antibodies. But there's definitely not 100% recycling. The other question is, what I've showed is that you have two pathways. You can either recycle back to the same membrane where you have the pinocytosis entry, or you have transcytosis. And the more interesting question is, how much does transcytosis contribute to the overall biodistribution? And that seems to be neglectable based on the experimental data that we have. So the majority of distribution appears to be through those pores, through the paracellular pathway, whereas the FcRn pathway is really for recycling back to the blood.
Yeah. There is a technical question on the system parameters. I don't know whether it's worth following up with this one separately, unless it's an easy one to answer. But it's, when you account for the tissue vascular volume, how do you calculate the venous and arterial volumes in your model?
The venous is 2/3 and the arterial is 1/3.
Okay, that was easier. Right. I don't see any more questions coming through. If anyone has a question that they want to raise themselves, you can always use the raise hand feature and unmute yourself if you had any question. Just give everyone. Yep, Jennifer.
Thank you, Felix. Thanks, Ollie. I was just trying to remember the 20%, the additional systemic clearance. Felix, you made a point that it's 20% of the total clearance. I mean, what was the top-down?
Yeah, so this is 20% of the total clearance that we set initially for IgG. Because IgG, you have like a lot of data exogenously where you know the PK very well.
Yes. So would that be kind of similar to actually fitting a profile if we have clinical data top down?
That's a very interesting question. I haven't tried this really. When you had the clinical data to just set it to 20% of that. The only thing I know is for all of the different cases we have used, it's not only those 7 that I presented today, but also other antibodies and antibody drug conjugates, is that the additional systemic clearance can be very variable between those molecules.
Yeah, that's why I was curious, is because we have examples where we have clinical data, but I was just curious if I straightforward used a 20%, how well predictive it would be for the same reason, because you said it's kind of variable from program to program slightly, but that's interesting. Thank you.
Okay. Well, I can't see any more questions coming through. Thanks again, Felix, for presenting today. I really appreciate you taking the time to walk us through your research and the collaborative work with the Biologics focus group. So I'm sure if people have any questions about that, they can also contact you. But otherwise, that's it for today. We're having a bit of a summer break, and we'll be back with more webinars later in the year. So look out for them in your mailbox. But thank you, everyone. Take care, and speak to you again soon.